Okay, welcome back to Chapter 21, Nucleic Acids and Protein Synthesis. Here we are in our sixth lecture, and we're back to a pretty short lecture again as we were for a few of the previous lectures. So we've done a lot now with DNA and RNA, and looked at those different processes of how the DNA replicates and then how the RNA can make sense of that in terms of coding for protein synthesis. Our particular focus here for this sixth lecture is the genetic code. So we'll go ahead and see in particular what those messenger RNA portions are looking for in terms of protein amino acids. All right, so I hope I didn't let the cat out of the bag too soon, but the genetic code is a sequence of amino acids in a messenger RNA that determine the amino acid order for the protein that is going to be synthesized. It consists of sets of 3 bases, triplets here, along the mRNA called codons, and it has a different codon for all 20 amino acids needed to build a protein. Again, we talked about the idea that there are 20 common amino acids for human proteins, and luckily nature figured out how to have a different codon for each of those amino acids. It contains codons that signal the start and end of a polypeptide chain, which is important, just as important as the sequence, right? Being able to start and end that peptide chain correctly. So if we look here at table 21.5, we have the messenger RNA codons, the genetic code for amino acids. And notice that there's some redundancy there. We have, for instance, UUU codes for phenylalanine, as does UUC. We've got UUA or UUG to code for leucine, and so on and so forth. We've got a lot of other leucine codes there that start with cytosine instead of uracil, so on and so forth. I don't expect you to memorize these. It's a lot, right? So please don't worry so much about having these committed to memory. Just understand that the body has specific codons that code for specific amino acids, and that you can have more than one codon for the same amino acid, but you can't have more than one amino acid for the same codon. That doesn't work. We also see the start and stop codons there that will initiate or terminate a peptide chain. Okay, so looking further at these codons and amino acids, suppose we want to determine the amino acids coded for in the following section of a messenger RNA. So we have the 5' end, we have CCU, AGC, GGA, and CUU, and then we're at the 3' hydroxy group. So according to the genetic code, we have our amino acids for these codons. The CCU would be proline, the AGC would be serine, GGA would be glycine, and then CUU would be leucine. So our mRNA section codes for the amino acid sequence as follows: prolynyl, serine, glycyl, leucine, if you remember your naming of peptides. So there we have it. The messenger RNA very discreetly and discriminately looks for those proteins with its codons. And believe it or not, we're already at the end of this sixth lecture, so we'll end with a learning check, as we often do. This time we're going to write the amino acids coded for by a section of mRNA with the following base sequence. And so there you see 5' end to 3' end. And notice, since I don't expect you to commit these to memory, rather than memorize all the possibilities, we have some possible codons listed down below. So that's what you'll be able to expect if you were asked a similar question like this come exam time. You're not going to commit everything to memory. That's just not reasonable. There's a lot in biochemistry to remember, and it just doesn't work well to try to memorize all of it. So there you go. From that list of possible codons, go ahead and tell me the amino acids coded for by that section of mRNA. Stop the video here if you need to. Start us back up when you're ready to check your work. Good luck. Okay, there you have it, and hopefully based on those possibilities, you were able to correctly identify this tripeptide as alanyl valine aspartame, or aspartic acid, or aspartate, depending on its form. So there we go. ALA-VAL-ASP would be the sequence. So not so worried about the name here, because that's not what you were asked for. So there it is. Hopefully it was relatively straightforward, especially since you have it there to draw from. So hopefully you're not so concerned If you think back to that fourth slide with all those possibilities, that may have been a bit daunting, but if I give you a reasonable amount of groups and you just have to pick them out, then hopefully it doesn't seem so bad. So if that's not the case, if you're frustrated beyond belief and none of it's making sense, then please do reach out to me. Otherwise, if things are going well, then we'll see each other again for the 7th lecture.